Oh my goodness, I am so delighted to be here. Thank you so much for that lovely introduction and telling everybody my secret name. It was Mumbai. I'm delighted to get to come to this magical place. This whole trip has been such an honor and just so fantastic for me. And I'm glad to have you as the finale of these talks. And so it's bittersweet for me to give the final lecture, but I sure am honored that I've been invited to be part of this monumental series and that you guys will come spend time to me to come learn about the science that I'm doing back home in New Jersey. So many, many heartfelt thanks from the start. My goal, which you've already heard a few clues about during this lecture, is to try to convince you that bacteria can talk to each other, to try to convince you that they're multilingual, to try to show you that learning about this ancient language tells us something about how multicellularity arose on this earth, but also has practical applications in terms of medical, industrial, and agricultural therapeutics or applications. And then in the spirit of the TNQ lecture series, I really want to focus on the basic discovery process. I think that you'll see as I go through the talk, you'll see, and I'll try to allude to some of the applications that are coming out of this quorum sensing field. But my real central goal for this talk is to try to remind you and convince you that we cannot have applications unless people make discoveries and deeply learn how biological systems work. And only through knowing those kinds of rigorous sort of understanding of biological systems can we then hope to make successful applications that are helpful to humanity. So it's really going to focus on the basic part of my gang's research. And then secondly, I thought what would be really interesting or hopefully interesting to you to do is to make sure that I give this talk starting from the very early history, which is in these bioluminescent bacteria, because this entire field of quorum sensing and this idea that bacteria can act in groups and can communicate with chemicals came from an obscure, harmless, ordinary bacterium that you never could have guessed would give rise to this field and to these possibilities for applications. And so I really want to try to make sure that I bring home the idea that studying something that you're passionate about and that you think is fascinating and that you have no clue could actually give rise to applications, that actually by studying those kinds of systems, you can actually move toward practical and useful applications for humanity. So that's sort of the theme of the talk, and let me get into it right now. So what my gang at the lab always wonders about is how bacteria get their power. So these are just pictures of famous bacteria taken under the microscope. The top set of pictures show very notorious bacteria that when they get in you or on you, or a plant or an animal, they can make you sick or they can kill you. Right? And so the question is, how can these puny, itty-bitty organisms have the power to kill or to make sick higher organisms that are so much bigger? And so for the 150 years or so, or the 500 years or so that we've known about bacteria, they've always been thought to be pathogens that make us sick. Increasingly, we're learning there's these other personalities that bacteria have, in that we now understand, and we've only known this for the past 20 or 30 years, that there's all these bacteria that live in and on every higher organism, and these bacteria are essential for life to happen. They're called the microbiome, this consortium of bacteria that live with different organisms, and what we now know is that these microbiome bacteria give higher organisms traits that our own genomes don't allow us to have. And so we're only learning about all of these characteristics of the microbiome, but a few of them we know. So for example, if you eat plant food, whenever you eat vegetables, you have no capacity to digest that food nor to get the calories and the nutrients from it. It's the bacteria in your gut that digest plant food and give you those resources. We know now that early in life, we become infected by bacteria and that somehow they train our human immune systems to know bad bacteria from good bacteria for the whole rest of our lives, right? By being there, educating our immune system and making it so we don't get autoimmune diseases. We also now know that microbiome bacteria give us all kinds of vitamins and micronutrients and things that make the components of our own bodies function. And so now you get it. These are facts. Bacteria can kill us, and we also know that they give us our lives. Those are known by scientists. What my gang is always trying to wonder about is how can they do either of these things? They're so simple, seemingly simple. They're so tiny. They're so ancient. How can it be that bacteria have the power to take our lives away and to give us our lives? And so, of course, what I want to try to convince you is that the way they do that is they communicate. The language is going to be chemical. And what they do is that they count their numbers, they act in groups, and if they carry out these tasks in synchrony, they can accomplish feats like these that they could never accomplish if a single bacterium acted alone, because individually bacteria are too small to make a difference. And so that's where we're going in this talk. And then again, I promised that I would tell you how this idea of bacteria acting in groups came about, how my field arose. And it actually came from this quirky animal, which is called the Hawaiian bobtail squid. So this animal is about this big, full grown, and it lives off the coast of Hawaii. And it lives in a beautiful one-to-one symbiosis with a bioluminescent bacterium named Vibrio fischeri. So Vibrio fischeri has the special property that it makes light. It makes blue light. And this squid and the bacterium have evolved this symbiosis because each of them gets something by interacting with the other. So first, let me tell you about it from the squid's point of view. So again, this squid, when it's full grown, it's just a tiny squid, but it lives just off the coast of Hawaii in just sort of shallow, knee-deep water, so just a couple feet of water. And this squid is nocturnal. So during the day, the squid sleeps, but then at night, it comes out to hunt. And it needs to be able to escape from predators both in the day and at night. And so first, let me show you something about the daytime behavior. So what we've done is we put the squid in an aquarium and the lights are turned on in the room. So the squid thinks that it's day and it looks very nervous, sort of spastic, because it's never supposed to be out during the day. If it was out like this, a predator would see it and eat it. So as I told you, the squid is nocturnal. So now what we've done, the lights are on, it's day, but we've given the squid some sediments. So it has spent a night hunting. Now it's time to go to bed. And so what you can see is it just camouflaged itself and it's going to bury itself in these pebbles, right, to keep away from predators. She's very thorough, right? You get eaten by a predator once, right? So what you can see is that she's covered herself completely with the pebbles. She's going to retract her tentacles in a second, and she's in bed. Okay, so that's a fantastic strategy. If I showed you this aquarium, you wouldn't know there's a squid in there. There's actually about 10 of them in there. Okay, so this is a really good strategy to stay away from predators during the day. She is invisible. The problem for the squid is that she's got to come out at night to hunt. Okay, so what happens is when at night, the squid emerges from this resting place, since it lives in just this shallow water, on bright nights when there's lots of starlight or moonlight, that light can penetrate the depth of the water that the squid lives in. So if the squid was just swimming around hunting and the moon was shining on it, the squid would make a shadow. And then predators could see its shadow, calculate the squid's trajectory, and eat it. So she needs a mechanism, the squid needs a mechanism to stay invisible at night. This is where this bacterium, Vibrio fischeri, this bioluminescent bacterium, comes in. So now you're looking at a squid that we've turned on its back, and I hope what you can see are these blue glowing lobes of this specialized light organ. So what the squid has evolved is an organ under its mantle or under its body. So it's a sac. And in this specialized light organ, this bacterium, Vibrio fischeri, lives in there jammed packed. It's like 10 to the 12th cells per milliliter of Vibrio fischeri. It's like toothpaste of Vibrio fischeri. So they're in that light organ and they're making bioluminescence. And so right under the body of the squid. And so what happens is when the squid comes out, it actually has detectors on its back. And so it senses how much starlight and moonlight is hitting its back. And then it opens and closes a shutter. It's just the squid's ink sac under this light organ where the bacteria are. So it senses how much light hits its back, and then it opens and closes the shutter. So the amount of light coming out of the bottom, which is made by the bacteria, exactly matches how much light hits the squid's back, so it doesn't make a shadow. It actually counter-illuminates itself using the light from the bacteria by matching it to the light from the sky. So now this squid has this fantastic strategy. It's invisible during the day by being asleep in the sediments, and then at night, this bacterium provides it this bioluminescence that makes it also invisible from predators. The bacterium gets something out of this. As I just told you, it's at 10 to the 12th cells per milliliter. It's all filled with gooey, goody nutrients inside this light organ. And so the bacterium can grow and make many, many more progeny than it could make free living in the ocean, because the ocean is a nutrient desert. So the squid makes more babies because it lives longer, and the bacteria make more babies because they get fed inside this light organ. Okay, so that's the partnership between the two organisms. But now let's talk about the Vibrio fischeri. It never makes light free living in the ocean when it's dilute. It only makes light when it's inside that squid at high cell number. So how does the bacterium know when it's out and when it's in? And that's by talking to each other. Okay, and so now what we did was to try to figure out about this bacterium talking to each other is we're going to use this property that it has of bioluminescence. So what you're looking at right now is just a picture of a flask and a couple of Petri plates and some ridiculous stuff that graduate students do in my lab, right? So we just drew that with a Q-tip, right? And we took this picture. To take this picture, we just turned the lights off in the room. This is what we see. We're not doing anything to the bacteria. They make this light, and we actually took the picture using the light made by the bacteria. And what was so fantastic about this obscure bioluminescent bacterium was this property of making light, because we could see it with our eyes. And what we could see is that when the bacteria were at low cell number, they made no light. But when they grew to a particular cell number, all the bacteria turned on light together. And that bioluminescence was super powerful because you can't see bacteria, with your eyes. You can't see them. You can't see their behaviors. GFP and genomes didn't exist at this time. So there was no way to track whether bacteria were doing something in a group or something alone. And so what was so neat about bioluminescence is we could just see it, we could measure it, we could follow it as the bacteria were doing this behavior of bioluminescence as a group. And so by doing that and using light as our readout, if you will, what we and the people discovered is that the way the bacteria understand times when they're alone from times when they're in a group, and in this light organ, for example, is by talking with chemicals. Okay, so this next cartoon shows the process of chemical communication. So this is supposed to be my bacterial cell, a Vibrio fischeri cell. So at low cell density, it doesn't make light. But what it does do is that it makes and releases small molecules that I just have depicted as these triangles that we call autoinducers. And you can think of them like hormones. So what happens is that the bacteria make these autoinducers, they diffuse away. The world is big, bacteria are small. When they release these autoinducers into the environment, the autoinducers diffuse away, the bacteria can't detect them, and that says don't make light. But as the bacteria divide and multiply, since all of the cells are making a share of these autoinducer molecules, the extracellular concentration of these chemicals is increasing in step with bacterial cell number. And when the autoinducers hit a particular threshold amount, the bacteria detect them, and they infer from that detection event that they must have neighbors around. And so in unison, all of the bacteria respond to the buildup of the chemical by turning on light. And so in fact, the bacteria have no idea how many other cells are around. They are simply measuring this chemical and using it as a proxy for cell number. And we know we're right about that because we can spin the cells out of the flask in a centrifuge. And if we take the liquids that the cells had been grown in and we squirt them on these cells, right, the molecules are there, these guys will turn on light, right? So they're simply measuring how much of that chemical is around, that chemical word. Okay, so that's how that works. And the question is, why am I standing on this stage? I mean, that's actually a deeper question. But one reason I think I'm standing on this stage is because this was originally discovered, as I just told you, in this harmless but beautiful bioluminescent bacterium that is not important industrially, medically, agriculturally, but it had that bioluminescence property that let us figure out how this works. And then if you fast forward a couple of decades, what we now get is it's not an anomaly. So this is the norm in the bacterial world. We now understand that bacteria must be able to distinguish times when they're alone from times that they're in groups and behave differently under those two scenarios. And the way they distinguish it is by talking with these chemical languages. And so now we have a name for this process, and we're a field. We call it quorum sensing. So the bacteria vote with these chemical votes, the vote gets counted, and then all the bacteria go along with this democratic decision-making process, and they carry out these group behaviors. And so in quorum sensing bacteria that have been well studied, somewhere between 200 and 600 genes will be controlled by these quorum sensing circuits. And so you might know that bacteria only have a couple thousand genes, and so a quarter of their genome is devoted to acting in groups. And so it is a big deal when a bacterium makes the decision to quit going solo and start to be part of a collective, and quorum sensing controls that process. And the other thing you might have heard about, and you heard about it in the introduction, is that now it's not all about bioluminescence, right? Globally important pathogens, agriculturally important bacteria, and industrially important bacteria turn on all kinds of traits. For example, pathogenesis, virulence factor production, biofilm production. They turn these traits on as groups. And so there's a huge amount of energy in the field to be able to manipulate quorum sensing. And so that is to make harmful bacteria not be able to talk to each other, to make good bacteria be able to chit-chat even better. And all of these applications are coming out of this quorum sensing field because we now know that these bacteria have to be able to act in groups for these tasks to be successful, right? And so the idea is when you make a biofilm, that's a community of bacteria on a surface. When you spew out your toxins in an enormous, eukaryotic organisms, if one bacteria or a few bacteria do them alone, those processes don't work. It's only when the bacteria get together, communicate, decide to do these processes in synchrony that these processes become effective. And so that is now quorum sensing. It's the norm in the bacterial world. I think the last time I looked in the databases, there were tens of thousands of cases now of known quorum sensing systems in bacteria, right? And so the applications are coming, again, from this sort of beautiful but harmless bioluminescent marine bacteria. Okay, so that's my introduction to quorum sensing. Of course, I want to tell you a little bit more deeply about how it works. The molecules are not triangles. They are chemicals, right? So let me tell you about the words that these bacteria are talking with. So we think that it takes at least 3 quorum sensing chemical words for the bacteria to have a proper conversation, if you will. And so what I've done on this slide is I put the molecules that this bioluminescent bacterium communicates with, right, as my examples. And so I'm going to tell you about those. But what I want you to understand is the principles I'm telling you under these molecules transcend all quorum sensing systems as far as we know, even though the molecules I'm using are from the original, the first bacterium discovered to have quorum sensing. So there's always a molecule. In the case of this Vibrio fisheri, there's this molecule, which is called a homoserine lactone. And as far as we can tell, one and only one species of bacteria makes and detects this molecule. So this molecule is for intra-species communication. This is how I count my siblings. The next molecule that we discovered, this fatty acid, we showed that all Vibrios make this molecule, but nobody but Vibrios make this molecule. So we think this is the language of the genus. So the first molecule says, you are my twin, you're my clone. The second molecule says, you're my cousin. You're not perfectly related to me, but you're closely related to me. The third molecule we discovered is broadly made across the bacterial world. They are all making the identical molecule, so there's no species information in this molecule. We think this is like the bacterial trade language. It's the Esperanto of bacterial communication. It's for interspecies communication. So now what we understand with these molecules is that what the bacteria get in communicating with quorum sensing is the first thing we think they do is they get information about how many cells are around them. But then what they get that encoded in these molecules is something about how closely or distantly I am related to my neighbor. So they both know something about number and something about who is in the vicinity. And in our recent work, what we've shown with those three molecules is that the bacteria actually measure the ratios of the molecules. And what we think they're doing is that they're asking, by measuring how much, say, of the self molecule, how much of the other molecule is around, they're asking, is it mostly me and my kin in this community, or is it mostly the enemy? And then what they do is they change which behaviors they carry out based on whether they and their family members are in the majority or their competitors are in the majority. So when they and their kin are in the majority, they share, they make all these public goods, they give away all these goodies to each other. When their competitors are winning because there's more of the non-self signal, then they try to kill each other. They make antibiotics, they make toxins, right? And they do very competitive behaviors. So what we get then is that they tell something about the cell number, but they also know, am I surrounded by my friends or surrounded by my foes? And then they act on that information. Okay, so getting even more deeply into this, what I thought I would do today is spend the rest of my time telling you about the sort of horrible cousin to Vibrio fischeri. The beautiful bioluminescent bacterium has a relative that you guys have all heard of named Vibrio cholera, which is a globally important pathogen that is endemic in different parts of the world. And you guys probably know that you get cholera disease by eating or drinking contaminated water. And so cholera is a pathogen, and quorum sensing controls pathogenicity in this bacterium. And so what we showed is that the way that it works is depicted on this cartoon. So when cholera gets into the human intestine, it comes in at low cell density, right? So these autoinducers aren't around, but under that condition, at low cell density, cholera is highly virulent. It's spewing out its toxins, right? It sticks to the human intestine and makes a biofilm. And under this condition, it makes the human being sick. And then you are basically a test tube of food for cholera. So as it grows and multiplies in the human intestine, when it gets to high cell number, right, and these quorum sensing autoinducers accumulate, when cholera detects those autoinducers, it shuts off biofilm formation, it shuts off toxin production, it regrows its flagellum, so it becomes motile, and then it cuts itself off the intestinal epithelium, and out it comes in huge numbers to infect the next patient. So cholera causes this acute disease. Its strategy is sort of comes in, guns loaded, get in, and then multiply like crazy and get out, and it's all about dissemination. That's the sort of trick for the disease. And so it must have quorum sensing, because quorum sensing controls these transitions, and it has to be able to do this repeatedly in the human host and in the environment. So quorum sensing is totally critical for those transitions to happen. And so once we discovered that it had quorum sensing, we wanted to discover, well, what are the parts that control quorum sensing and virulence and biofilm formation in Vibrio cholera? And so we did a lot of genetic screens to figure out the components. And on this cartoon, I'll just show you some of the key players. So cholera has receptors on its surface to detect and bind those autoinducer molecules. And at low cell density, when the molecules aren't there, these receptors turn on a set of small RNAs. And so for those of you who work on eukaryotes, these are the microRNAs of bacteria. So they're small RNAs that regulate gene expression. So these small RNAs get turned on at low cell density, and then they go on to turn on biofilm formation and toxin production, right? So that tracks with what I showed you on the last slide. At low cell density, cholera is super virulent. Then at high cell density, the autoinducers accumulate, and cholera only uses 2 of those 3 autoinducers that I showed you before. And so they actually have names that you don't need to learn, but these molecules accumulate, they get bound by the receptors, and when that happens, it flips a switch, and the receptors stop allowing these small RNAs to get made, so biofilm formation is repressed, virulence factor production is repressed, and cholera flees the current host in high numbers to infect the next patient, right? So this is sort of the ins and outs of that cartoon that I showed you on the last slide. And so graduate students in the lab had discovered and characterized these different components. And around the time that we were working on these and trying to understand, how all of this happens at the bottom of this cascade, a fantastic postdoc named Kai Pappenfort came to the lab, and he was thinking about the regulation of this set of genes, the biofilm genes and the virulence genes. And so he was doing genetic screens to look for regulators. And what Kai found was new genes that we had never discovered in this quorum sensing circuit to that date. And so he discovered a gene and a protein that he named VQMA for Vibrio quorum A, like A, first letter in the alphabet, as if I hadn't existed for the last 20 years, right? He's got the A protein. So he finds this protein, okay, you guys are allowed to laugh. Anyway, this VQMA protein, right? And it's a transcription factor, right? So it's a DNA binding protein. And then what he found was that it controlled another small RNA that he called VQMR for RNA. And then at high cell density, this circuit fed into the circuit we already knew to shut down all of those genes and allow cholera to flee from its current host. And so that was curious to us that there seemed to be more genes and proteins involved in this circuit. And so then what Kai wondered is, how does it work that this small RNA only turns on at high cell density? Because what was curious was this protein VQMA is always present in the cell. It's constitutively produced, its amount never changes, so it's always there, but it only functions at high cell density. And so what that meant was that there had to be something that was regulating the activity of VQMA, not its production, its activity, so that it would only work at high cell density. And so when we see some protein whose activity is controlled at high cell density, in my lab, we always go, oh, that's got to be quorum sensing, right? And so maybe our autoinducers that we had already discovered are also controlling this new circuit. And so we can make these autoinducers in test tubes. We have mutants of cholera that don't make one, the other, or both. And so we tested every which way we could. Like, did we add this molecule and turn it on? This one, this one, this one. Nothing we added changed the function of this circuit. And so what we had to infer from that experiment that was an utter failure was that we were missing an autoinducer, right? There had to be some other autoinducer that we had never found that presumably interacts with this VQMA protein at high cell density to tell it to turn on this small RNA and feed into the circuit that we had already known that controls these hundreds of genes that are required for cholera to be a pathogen and get in and out of the host. And so Kai, of course, had discovered this part of the circuit, but he didn't know what the molecule was. But he loves small RNAs. And so he left to start his own lab in Germany. So he's a professor in Germany, and he works on the small RNA part of this circuit. But what was maybe good and bad was that a young first beginning graduate student named Justin Silpe had come to the lab. He was a first-year graduate student, and he was doing what's called a rotation in my lab. So at my school, when graduate students come the first year, they try out like 3 or 4 labs for 8 or 10 weeks, and so they can decide which lab they want to do their PhD thesis in. And so Justin was trying my lab out. He was involved with Kai in discovering the VQMA and VQMR circuit. And so he thought that was fun to do because they had made a discovery. And so Justin decided to join the lab, right, and become a graduate student in my lab. And so since he had already been working on this part of the circuit, of course, the big mystery was, what is this molecule? And so Justin decided, as part of his rotation, to take on the project of figuring out what this putative new autoinducer might be. And so that's when Justin made his first really important, for this part of the story, his important discovery. So he was trying to understand the role and the structure of this molecule. And what he noticed was that if he just collected cell-free supernatants from all kinds of different bacteria and added them to Vibrio cholera, this circuit would turn on. And so his interpretation of that was that this was going to be one of these broadly made, perhaps generic autoinducers, right? Because lots of bacteria seem to make the activity. And that included laboratory E. coli. And so he thought he could use laboratory E. coli, because we can grow it and we can manipulate it really easily, he could use it to be able to figure out the structure of this putative molecule. So here's the experiment Justin decided to do. So he took regular old lab E. coli and he grew up a couple of, liters of E. coli. And then his idea is E. coli is naturally making this putative new potential autoinducer molecule. And so into E. coli, he cloned the cholera VQMA gene. And he had it under a high expression vector, so E. coli was pumping out tons of this protein. And he put onto this VQMA protein what's called a his tag, right? So it's a bunch of histidine residues, and they bind to nickel. And so what he did then was he had the only cholera gene, the only cholera protein, none of this other mess was there, just VQMA was there. He grew a bunch of E. coli, and then his idea was E. coli would provide this putative autoinducer, it would bind to this VQMA transcription factor, and then what he could do is he could just mush up the E. coli and then take the lysates and put them on nickel beads. And so the nickel beads would grab on to this VQMA protein, and he could wash away all the other E. coli proteins. So his idea is that the protein is stuck there and that the ligand, the new autoinducer, might be stuck on that protein. So then what he did was he just heated these beads up to like 60 degrees Celsius. So he heats them up and the protein unfolds, and his idea was then it would drop this autoinducer out of the protein. He could just spin the beads and the protein out in this Eppendorf, and what would be left over in these liquids would be his molecule. And so and he made this ridiculous animation. Did you guys see that? Yeah, now you're getting an idea what Justin's like. Okay, so then he could just collect this up, right, get rid of the protein, and then we could take this across the street to the chemistry department, and we could do mass spec and NMR and figure out the structure of the molecule. And sure enough, that's what Justin did. And it turned out that this was the mystery molecule, the new autoinducer, and we call it DPO, and that just has to do with its real chemical name. But what you can see is it's a really simple molecule. And what I'm going to tell you right now is that the left-hand part, this side of the molecule, that's made from the amino acid alanine. And the right-hand side of the molecule, that's made from the amino acid threonine. So two amino acids come together to make this molecule, and that is a brand new molecule to mankind. So it had never been known before until Justin discovered it. Okay, so now we were kind of cooking with gas, right? Now we had this new molecule, DPO, that works in conjunction with the ones we knew about, and all of these systems or these molecules come together to tell cholera it's time to leave the current host and infect the next patient. Okay, so if I put together this part of the talk, what we had learned so far, and what I hope you're learning, is that cholera, when it gets into the human host, it has to decide, should I stay or go, stay or go? Quorum sensing mediates that decision. And so a number of quorum sensing molecules are involved, but in the news story, this new molecule that Justin found, DPO, accumulates as cholera grows, the VQMA protein binds it, and it shuts off pathogenesis and turns on the escape genes. But you're going to remember I already told you that Justin found that many bacteria make this DPO activity, and that includes microbiome bacteria. So it turns out that these microbiome bacteria are making buckets of this DPO in the human intestine. And you're also going to remember that I told you it's made from threonine and alanine. So it turns out that, and Justin showed this, is that no bacterium will make DPO using its own threonine. Bacteria can make threonine, but they won't use it to make DPO. They have to get the threonine from the external environment. And so it turns out you're the ones that give them that. So these are supposed to be your intestinal epithelial cells. Your cells are covered with this gooey protein called mucin that keeps everything hydrated and keeps your microbiome bacteria away from your human cells. And microbiome bacteria have mucinases. They sort of eat mucin for a living. So you provide them this mucin, they eat the mucin, and they grow happily, and that keeps everything in harmony in the gut. It turns out that every third amino acid in mucin is threonine. So this is an inter-kingdom conversation because you, the human host, provide your microbiome bacteria the threonine that they must have to make DPO. And then they make a lot of DPO, and what happens when DPO is there? Cholera disperses. And so what happens, depending on the blend of microbiome bacteria that are in the gut, and if there's a lot of bacteria that make a lot of mucin, cholera miscounts and it flees too early. And so it's known in humans that some humans get severe cholera, some humans get not so severe cholera. We haven't proved this yet, but what we think that might have to do with is who is in the microbiome and how much of this DPO is being made or not. And so that has been shown in a mouse, right? Is the DPO is mediating how long cholera resides in the intestine. And we're wondering then if that might be true in humans. And if so, we can imagine giving humans DPO, or even perhaps easier, giving them threonine, right? And that could be used either prophylactically or perhaps as a medicine in order to fight against cholera disease, right? So that remains to be seen. But what we know for now is that there's this new DPO molecule, and that we think that this is a multi-species, multi-domain conversation in which the human host has a part, the microbiome has a part, and the predator, cholera, also has a part. Okay, so that's where we got so far, and Justin, I told you, discovered that molecule. What I also told you was that Justin was a rotation student, and so I'm hoping that, like all the graduate students, there are graduate students here, right? Oh, good. Okay, good. Anyway, so I'm hoping that you guys think I'm really friendly and I'm nice, and wouldn't it be great to have me as your advisor, right? And so anyway, so Justin was in the lab 10 weeks. I'm not letting him graduate. I was like, get to work. What are you going to do next, right? And so anyway, he decided to join the lab, right? And then he had to figure out his next project for his PhD thesis. And so, of course, you guys are already going to be able to guess that he wants to work on this system, right? And so I've already told you the first hint that he had made about how he was thinking about this. So you're going to remember that I told you that Justin showed that DPO is broadly made in the bacterial world. Tons of different bacteria make DPO. But what was weird about that finding was that he also looked at VQMA, right, the transcription factor that binds DPO. It turns out that VQMA is a Vibrio protein. Every Vibrio, every sequenced Vibrio has a VQMA. It's always intact. There's never a mutation or a stop codon. But no bacterium except Vibrios have VQMA. So there's this strange asymmetry that we've actually never figured out, because we got distracted with what I'm going to tell you next, between apparent production of the molecule, which is broadly spread, and apparent detection of the molecule, which is extremely restricted. Now, it could just be that other bacteria have some other receptor, or maybe they don't detect DPO, but Justin was hunting around trying to understand this weird pattern. So he's looking in all these databases to try to make sense of it. And so I told you that he could never find another bacterium that made VQMA, but he did find one more VQMA. And the curious thing about it was not in a bacterium, it was in a virus. And so viruses that infect bacteria are called phages. So just like we are bombarded by viruses, bacteria are bombarded by viruses and all the time, but we call them phages. And so what Justin found was there was a genome sequence in the database of this phage. So some scientists had been making collections, and they had collected this phage, they had sequenced it and put it into the database, but they had never studied it. And so what Justin saw was on this sequence in the DNA database, there seemed to be this phage that has a name that had a gene on it that encoded a VQMA receptor. And before that moment, quorum sensing was supposed to be about bacteria. It's not supposed to be about viruses. And so we wondered what the heck was going on. Could there be a connection between quorum sensing and viruses? And so what we did was we got this phage into the lab, and Justin started to study it. And so before I tell you about this, I want to turn your attention to this right-hand part of the genome. So there's lots more genes on this phage, but there were genes that we could recognize from classic studies of phage lambda that have been done 70 or 80 years ago. So you guys probably know that lambda and studies of phage lambda founded, the molecular biology revolution, right? And on our phage and also on lambda and many other phages, there's two important genes. One encodes a protein called C1, one encodes a protein called Q, and then there's a whole bunch of genes that the bacteria need to kill the host. So phages like our phage and like lambda are what are called lysogens. So they can undergo one of two lifestyles. They can live in their host and just be passed down. They're called temperate phages. Anyway, they can live in their host and just be passed down through generations, and that's called lysogeny. Or they can replicate like crazy, kill their current host, and spread to the next host. That's called lysis. So temperate phages like lambda and like this phage have to decide between lysogeny, lysis, lysogeny, lysis. C1 and Q mediate that decision. And so the way that it works in, for example, lambda, is that the phage will merely live in the host as a lysogen, just living dormantly until the host gets in trouble. So whenever the host is under stress, if there's DNA damage or SOS, the phage says, that's it, I'm out. And it starts replicating like crazy, kills its host, and tries to get out of the host that's stressed and get into another host. And so what happens is when there's DNA damage, this repressor called C1 gets cleaved, that inactivates it. The Q protein gets made, and it turns on all the lysis genes, and lambda flees. Exactly that happens in the phage that Justin was working on. He copied the exact classic lambda experiments, and it works exactly like in a textbook. Okay, so we could understand the right-hand part of this genome. Our question wasn't over there. Our question was here. Why is there a VQMA gene, a receptor for a quorum-sensing gene on a phage, and why is our quorum-sensing gene on this phage? And so Justin wanted to study that. And so I told you he had just discovered DPO. We just knew from Kai about the receptor. And so we thought, well, what experiment can we do? Well, we only knew how to do one experiment. Let's just add DPO and see what happens. Okay, so that's on this next slide. What Justin did was he infected Vibrio cholera with the phage. And now what you're looking at, this is really simple. This is just a growth curve of Vibrio cholera that's carrying the phage. And what you can see is it grows just fine. Right? Couldn't care less that phage is living, in its cytoplasm. But if Justin adds DPO, the autoinducer, now look at the white circles. All the cells die. And so what we think that means is that the phage is eavesdropping on host quorum sensing, right? It's recognizing when the host is growing to high cell number, and it chooses to only kill the host when it's at high cell density. And if you think about it from the phage's point of view, right? When a phage decides to pop out of its current host cell, if there's no other cells to get into, that phage is a goner. So when's the optimal time to leave your current host? Well, it's when there's lots of other hosts around. And so we think that what this carrying of this receptor does is it allows the phage to eavesdrop and surveil the cell density of its host, and it exclusively kills the host at high cell density, and that maximizes transmission of the phage to its next host cells. So it's got lots of victims around, and it's the optimal time to kill. And so we were really interested in that because for the 70 years or 80 years that it's been known about phages and lambda, all phages, it's always been thought that the only signal, the only information phages tune into to decide to do this lysogeny lysis transition is host cell SOS or DNA damage. And so our phage does that, but what we think is that perhaps phages can tune into other signals that the host is making, in this case a quorum sensing signal, and use that information to time these lysis lysogeny decisions, right? So that's what we think this connection to quorum sensing is. And so then the next question for Justin is, okay, we have this classic lysis lysogeny machinery, and now we have this phage quorum sensing system. How are these two circuits connected, right? What is connecting phage eavesdropping to what we know, these components that are required to flip that switch for the phage to lyse the current host? And we thought, oh, this is going to be easy. So you're going to remember this is a transcription factor, VQMA, right? It binds DPO, it binds DNA, and then it turns on gene expression. So we thought, well, how easy is that? It's going to bind in front of Q at high cell density, turn it on, and the cells are going to die. So Justin tested that, totally wrong. He said, that's okay, we'll just move upstream. A repressor of a repressor would also work. So it must bind at C1 and turn it on, let it get going earlier in the system. And so Justin tested that, totally wrong. And so now we were out of components that we could understand. And so Justin had to infer that there must be some other missing mystery component that connects this circuit and links phage eavesdropping to the classic lysis lysogeny decision-making machinery. So to try to find that, what he did was he did a genetic experiment. He just chopped up the phage chromosome and cloned it in with this stuff into a bacterial cell and looked for lysis to happen. And sure enough, he got one piece of the phage genome that was required for that. It was exactly one gene that he was missing. And it was right here. It was this gene that I have depicted in red that was sitting right next door to the VQMA gene, but counter-oriented, transcribed in the other direction. And so this gene encodes a 78 amino acid protein, so a tiny, tiny, tiny little protein, 78 amino acids. And when Justin took the sequence of the gene, the sequence of the protein, and he put it into the database, it matched nothing. So this was a brand new gene of unknown function that made a brand new protein that had never been known before. And because that was the case, that meant Justin got to name it. It's his gene, his protein. He gets to give it the name. And so he calls it Q-tip, because Justin does not care about Bonnie Bassler's reputation. So he says, Bonnie, no, you understand, it's obvious, he says to me. This means quorum-triggered inactivator of C1 protein, Q-tip. And I said, Justin, that does not roll off your tongue, right? That is not obvious. But of course, I've already told you his gene, his name. So this little connection protein is called Q-tip. All right, so then the task was to figure out what is Q-tip doing to connect these two sets of genes on this phage. And so Justin did lots of experiments. I'm just going to tell you the one that's important, that worked. So what Q-tip does is it sequesters that C1 repressor. Okay, so I'll show you the experiment. So what you're looking at here, you're just looking at a picture under a microscope. This is E. coli, laboratory E. coli, that Justin has cloned C1 from the phage into this E. coli. And he put on it what's called a halo tag, so that makes it green. And so what you can see is that all of the C1 is diffused through the cytoplasm. It's just in the cytoplasm, spread out uniformly. But then if Justin also clones in Q-tip, his new gene, along with C1, what you can see is the Q-tip gloms on to C1, it pulls it into these big oligomers, and they drop out at the poles of the cells in these big, in glop, right? And so if you look at that, they look like Q-tips. Okay, so that's actually how the protein got its name. It makes the E. coli and the Vibrios look like Q-tips, okay? And its job is to sequester this C1 repressor. And so the other experiment that Justin did, it's not shown here. C1 here is active. It works as a repressor. C1 here is inactive. So this sequestration mechanism kills the activity of C1, and then the cells go on to lyse. Okay, so now what I've shown you is that there's actually two systems and two inputs that control whether or not this phage is going to be a lysogen or it's going to lyse its host. The first one's classic, comes straight out of a textbook. When the host is in trouble, when there's SOS or DNA damage, C1 gets cleaved, Q gets made, and the phage kills the cells and moves on. The next one is new. It's quorum sensing, it's eavesdropping. The phage carries this receptor, VQMA, and so when the cells grow to high cell density, VQMA binds DPO, and then this complex does sit at the Q-tip promoter. Q-tip gets made, Q-tip gloms onto C1, pulls it out into these big oligomers at the poles of the cells, which inactivates C1, Q gets made, and all the cells die, right? So the fate of C1 is different. In one case, it gets cleaved. In the other case, it ends up in those oligomers. But the outcome to the cell is the same. It gets killed, the phage is replicated, and it moves on. Okay, so there's two inputs to lysis. But if you remember from the beginning of the talk, there's also two quorum sensing systems running here. So remember, the host has quorum sensing as well. So you're going to remember from Kai and Justin's work, the host has its own VQMA protein. It's also monitoring the buildup of DPO, the autoinducer. At high cell density, it turns on that small RNA that then turns on hundreds and hundreds of host genes that allow Vibrio cholera to change from being a pathogen to escaping the host. And what Justin showed is that this complex also binds the host promoter. So while this phage is getting ready to execute its host, it also messes around with hundreds of genes and, with all of the host biology. And we actually don't know what good it does the phage to be able to control all of these quorum sensing genes. We just know that it does that. The reverse is not true. The host VQMA does not bind at Q-tip. that should make sense. If it bound there, it would turn on the lytic program and it would kill itself, right? And what we did was we crystallized both of these proteins with the DPO bound, and we could show that there's a 2 amino acid difference in the DNA binding domains between these two proteins, and that allows this protein to be promiscuous, but it makes this protein be very restricted. So we understand the molecular mechanism for how that works. This one binds multiple DNA binding sites, and this one binds just one. Okay, so we still don't know the biology under this, but we understand how it happens. Okay, so when Justin got that far and we thought we were learning something about the mechanism and this new eavesdropping strategy, we got really worried that this was just some one-off in evolution, that somehow this phage picked up this gene and that it was an accident of evolution. And so we wondered, is this really a one-off or not? And so what was really lucky was by the time Justin got this little module, right? And what I mean by that is a gene for a transcription factor right next to a tiny little gene, in this case Q-tip, he could take this 2-gene module and search the databases and ask, are there any other cases of this? And lo and behold, sure enough, we have found hundreds of phages, all of which are in the genome database, none of which have been studied, but they all have a common module. And what I mean by that is they all have a gene that encodes a transcription factor, and then always sitting right next door is a tiny little gene running in the opposite direction that encodes an anti-repressor. And so each of these transcription factors binds its own stimulus, but they work in partnership with these Q-tip-like anti-repressors to control these lysis-lysogeny decision-making. So now what we think is that this is not a one-off. In the case of the phage that Justin studied, the phage is listening into DPO, to host quorum sensing, and it's taking that information and it's connecting it to its lifestyle changes using this Q-tip mechanism. These other phages all have a transcription factor. The transcription factor always has what's called a pass domain, which is for binding a molecule, but we have no idea what these other stimuli are, what information these other phages are listening into in their hosts. But what they presumably do is that they take in some appropriate host biology and they connect that biology using these new little anti-repressors to these lysis-lysogeny decision-making genes. And so we think then that it's going to be very common that is for this sort of phage host interdomain crosstalk, right, that allows these phages to be much more plastic and versatile and to take in more information to control whether or not they stay in the host or whether they go, right? So we think that this is a cross-domain communication mechanism as well. Okay, so now what I've told you is that these phages and the bacteria are talking across with these molecules. And then once Justin got that far, we thought, well, wait a minute, maybe there's actually even more to this. Maybe beyond talking to your host, maybe these modules also allow phages to interact and to compete with one another. And so here's our newest experiment that I'll show you that we've been working on. And let me tell you what I mean by that. So most bacteria in the biosphere are what are called polylysogens. And what I mean by that is that they naturally carry around many, 2 or more phages. And so that includes like E. coli K12. E. coli K12 is riddled with phages. It has all these phages in its cytoplasm and in its genome. And so what I also now know from Justin's work is that some of these phages have these new 2-gene modules that I've been telling you about. And so, for example, I told you about, Vibrio cholera that had a phage that has, this transcription factor and Q-tip on it. I told you that that's not an anomaly. We've found many phages that have a transcription factor and an anti-repressor that's like Q-tip. Now what we've found are bacteria that naturally harbor 2 phages, each of which has one of these 2 genes modules. They're not the same 2 gene modules, but they each have this new mechanism on their genomes. And so what Justin wonders is what happens when you have a setup like this. And not only are these phages listening into the host, but maybe they're using their modules to compete with one another. Okay, so here's the last experiment where we're trying to figure this out. Okay, so we have a bacterium, right, that now, like as I've told you, is carrying two phages, right? It naturally carries two phages, and I just put a little part of the genome here. Each phage has its own transcription factor gene, and it has its own anti-repressor gene. They each have their own C1 repressor, right? And so now what happens is if we subject that cell that's carrying these phages, right, to DNA damage, the classic textbook signal that makes the switch from lysogeny to lysis, if you subject it to DNA damage, what happens is that the C1s both get cleaved. I'll call them the pink one and the blue one, right? The phages are like, I'm out of here. They both replicate. They make many more phage virions. They kill the current host, and they get out into the world. And what we can do then is we can just collect up what comes out and count using sequencing how many of the pink genomes are there and how many of the blue phage genomes are there. And so if we monitor how many of there are, these are just how many sequencing reads, what you can see is it's a tie. It was a race. The host got in trouble. Both of these phages induced their lytic processes, and about 50% of the viruses that came out were the pink one, 50% were the blue one. So they're just racing to the end, and they're about half and half. But what if we do a different experiment? What if instead of giving the general DNA damage cue, we give a specific cue? We give the input, the signal that is bound by this top transcription factor. So if we do that experiment and we add the signal, now this transcription factor binds its ligand, it turns on its Q-tip. But this transcription factor on the other phage doesn't recognize this signal, so it's not doing anything, right? So now what happens is that this C1 gets made into oligomers. This C1 is perfectly fine. This phage induces its lytic cascade because it's done its eavesdropping, but this phage didn't. And so now if we collect up the soups and count the phages, there's 100 times more of the pink virions than there are the blue ones. So this phage propagates like crazy, and this one lost the race. So when the cell gets killed, there's hardly any of these phages to go on to the next host. And of course, we can do the opposite experiment. We can add the ligand for the blue phage. So now this phage, C1, gets sequestered. This one is inert. It induces, and now what you can see is that the outcome of the phage-phage competition is exactly the reverse. In this case, the blue phage wins and the pink one loses. So what we think then is that in addition to allowing these modules, in addition to allowing phages to interact with their hosts, they are absolutely dictating what is happening in terms of the phage population and the host bacterial population. Okay, so that's our newest experiment to try to understand how these things work in nature. And so now I'll try to sum it all up, starting with the bacterium Vibrio cholera, right? This terrible pathogen, when it gets into the host, it has to decide, should it stay in the current host or should it move on to other hosts? Quorum sensing is going to control whether cholera stays or goes. In this case, this new molecule DPO plus the other autoinducers I show you, they accumulate at high cell density. Cholera shuts off its virulence repertoire, and it leaves in high numbers to infect the next patient. You, the humans, are helping out in what's going on in there by providing your microbiome bacteria threonine. Your bacteria, your microbiome bacteria make lots of DPO, and that confuses cholera, makes it miscount, and cholera disperses early. So this is a three-way conversation between the human, the good bacteria, and the pathogen. But then, of course, there's more to it than that. Now what we know is that there is the parasite of the parasite. And so now we know these phages are also monitoring the buildup of that DPO molecule, and they're making sort of a similar decision as cholera. They're asking, should I stay in my current host or should I flee? In this case, the current host is a bacterium. And so this DPO molecule is dictating when the phage is going to stay or go, but it's also going to be killing the cholera, and so that will be changing the numbers of cholera in the intestine. And then, of course, under these conditions, there's going to be DNA damage, and if there are polylysogens in there, they're going to be coming out of their current hosts, and we can imagine that these other stimuli that work with these modules are also there or not there, and all of this chemical information and sensory information is dictating who's winning and losing, right, in these complicated consortia that include beneficial bacteria and pathogens and viruses. And so the goal for my lab now is to sort of up the complexity of our experiments. We're trying to put together experiments instead of, one bacterium and one phage or two phages, in a test tube. We're trying to gin up experiments that look a lot more like this so we can really understand how anybody gets robust information out of these molecules and figures out what to do and when, given that all of this is happening and more is happening simultaneously. And so that's sort of a new horizon that we think is an interdomain eukaryotic bacterial viral conversation, and we're trying to figure it out. And so just really briefly summing up the high point, what I hope were high points to you, is that the bacteria talk to each other and their language is chemical and they are multilingual. There are many molecules, and I've showed you the newest one involved in this lexicon. What quorum sensing lets bacteria be is multicellular. They can accomplish feats that they could never accomplish as individuals by acting as groups. And we think that the rules of the game for doing this robustly perhaps are involved in what happens in higher organisms. Given that we all came from bacteria, maybe some of the principles are embedded in these bacterial quorum sensing systems. And by studying this, we'll be able to help our colleagues who try to think about multicellularity in higher organisms. And of course, with these chemicals, bacteria can distinguish self from other, friend from foe. And of course, that's what the cells in your body do. It's not like your heart cells and your kidney cells get mixed up every day. And that's because they use different molecules so that they can take on different fates and carry out particular jobs. And again, we think the principles of that are in these quorum sensing systems as well. And then, of course, I hope the main point for today in our newest work is that quorum sensing, until a couple of years ago, was always thought to just be about bacteria. Now we know that the eukaryotes and the viruses are involved, so we think that quorum sensing spans across domain boundaries, and we think this is new territory that's ripe for study by scientists. And then, of course, there's all kinds of applications for medicine and for agriculture and for industry. I told you, we try to make pro and anti-quorum sensing molecules. Maybe we can do something with DPO and threonine. Justin also made a bunch of phage therapies that we can add the stimulus and the phage kill on demand. And so we think that there may be quorum sensing in therapies across these different kinds of strategies. And we work on trying to stop the bad bacteria from being harmful and perhaps improve quorum sensing in the beneficial bacteria. And a lot of people in my lab work on those kinds of applications. And then the last thing to do is to finish with a confession, that we've thought this up in the lab over the last 20 or 30 years. Of course, the bacteria thought this up billions of years ago. And so if you go out in nature, you can see that these bacteria are, free riding and cheating and eavesdropping and making misinformation, and they eat each other's autoinducers, and they're doing all of these shenanigans. And so there's a lot of people now working, or scientists working, to try to sort of go out and do bioprospecting and try to find these naturally occurring pro and anti-quorum sensing strategies, because those have been tested over billions of years of evolution, and we should bring those into the lab as the building blocks and tinker with those, because these could prove to be the best when we really try to think about applications. And so that's where we are. And let me finish. There's Justin for the graduate students. So he's still alive. Yeah. And for the graduate students in the audience, he did get a PhD. Anyway, so Justin, he did all the work except the beginning work that I told you about Kai. And so he was a terrific, and I should tell you for the graduate students that he had never had a biology class when he came to my lab. His undergraduate degree was in engineering. He didn't even know what DNA was, right? And so I hope that makes you think that you can just do anything. If you're interested in it, you can learn it, right? And of course, he didn't work alone in my lab. So here's my gang. That's Justin back there. Here's my gang. That's in my front yard in Princeton, New Jersey. And of course, I had to pick a project to tell you about today. But every one of these people has an equally fantastic, in my view, equally fantastic project. We're quorum sensing. We love to work together, and the group gets more than the individual. And so they all work together. And of course, what I hope you see is that they look just like you, right? They're all undergrads, grads, and postdocs, right, that are changing the face of my field, changing the way we understand understand nature by making discoveries, fundamental mechanistic discoveries about how nature works. And then, of course, our grand hope is that by doing that, by just being curious and learning how, in our case, bacteria and their viral predators work, we hope that we will make information that is useful to other scientists whose sweet spot is to make applications, right? But you have to build those applications on discoveries, and those discoveries get made by undergraduates, graduates, and postdocs. And so they are just like you. And I want to thank TNQ. It is one of the greatest honors of my life to get to come to this magical place and give this series of seminars. So I thank especially Mrs. Ram for inviting me. I thank you for listening to me, and I'm really delighted that I got to come to this institute. So thank you for hosting me today.